So it gives me great pleasure to introduce my fellow colleague at UC Irvine, truly outstanding scientist. I had the pleasure of meeting her when I was a postdoc back in 1992. She's a professor of molecular biology and biochemistry, also pathology, and also in the Department of Neurobiology and Behavior. Almost since the first time I met her, she has been advocating for the role that inflammation plays in Alzheimer's disease. And a lot of recent genetic evidence really points to the fact that what Dr. Tenner has been saying for so long is true. A recent genome-wide association study has identified about 10 new genes that increase your risk for Alzheimer's disease, and many of those genes are somehow related to brain inflammation. And so when we were thinking about what we should present at this conference in terms of how it relates to the National Alzheimer's plan of trying to slow down this disease by 2025, we thought about new therapies, and obviously inflammation is a cornerstone there. She has received many accolades throughout a very distinguished career. She is also an elected fellow to the American Association for the Advancement of Science. She served as the Associate Dean for Research for the School of Biological Sciences, and most recently is the President-elect of the International Society on Complement. And she'll describe exactly what that is during her talk. And I don't mind telling you that she is my go-to person when I need support in trying to move forward an initiative. She's always incredibly positive and very eager to work hard to help advance the Institute and UC Irvine. So it gives me great pleasure to introduce Dr. Andrea Tenner.
Well, first off, thanks very much to the organizers, and it's a real privilege to be able to address this audience and to try and convey to you what some of the things we are doing to try and address this disease and get novel therapeutics that are going to work. Okay, so let's sort of get started on this. And one of the things that One of the things that I wanted to tell you about is what we're going to end up with is perhaps what's called a cocktail approach to AD therapy. Just like you have for heart conditions where there's statins, diet, exercise, I think there's going to be a cocktail approach as there is for HIV, concepts that we're borrowing from these other diseases to therapies for Alzheimer's disease. And these are going to be able to come into play perhaps at different points in time and stages of the disease as well. So let's begin. And as you might have all seen this before, but this is a stain of a human brain, and these are these amyloid plaques that now Dr. Flesher is showing you can be imaged in live individuals. This is obviously in a deceased individual, and these black arrowhead-like structures are tau structures. So these are dead or dying neurons. And these are the hallmarks of Alzheimer's disease. And you can again see the shrinkage with these dying neurons, neuronal loss, dying neurons, happens. You get these big fissures and less brain material in the Alzheimer's disease brain versus that of the normal individual. So this neuronal loss then is what is actually leading to the dementia. And what we want to do is try and begin to figure out when can we attack this and how can we attack it, and can we attack this disease with different reagents at different times. And so as everybody has talked about in the past, there's these preclinical phase of the disease. This is a slide taken from Claudia Kwas, my colleague, a long time ago, modified it a bit. And there's initiating factors, not real sure what those initiating factors are. There's factors clearly that promote it, as Dr. Flesher has said before. There are different factors in individuals that are going to determine the rate of amyloid accumulation and how that brain handles that amyloid. And that's one of the things I'd like to get into today. And so then what happens is obviously the catastrophic phase where you get this fibrillar amyloid coming in, which is what the PET scanning is seeing. And what I'm going to talk to you about today is this complement activation. And I'll define that in a few minutes. And we believe that that is really increasing and accelerating the progression of the disease and promoting the cognitive loss that is really what is so devastating in this disease. So it's sort of an overview of my presentation then would be to try and define for you what we mean when we talk about neuroinflammation and how it's different from other kinds of inflammation, and how this complement cascade, or this form of the immune system, is an inducer of Alzheimer's disease neuroinflammation, not the only inducer, but one that we can attack. And that while some of these components are detrimental, some of them are preventative and provide protection. And so when we're treating these, we don't want to just inhibit everything. We want to be strategic about it and not inhibit the protective or reparative functions. And so all of these components put together, we want to be able to identify what specifically are the components that we can address and target for therapeutics. And how do we go about doing that when we're starting very early on in the basic research arena? Okay, so first off, I'm going to take you a little bit aside to the immune response. That's where the inflammation is coming from. And the immune response's goal is really the elimination of pathogens and getting rid of cellular debris. And everybody thinks about the immune system when they think about antibodies. And if you're just a little bit more sophisticated, you might think T cells. Those are part of what's called the adaptive immune response. But that's clearly only part of the immune system. And the one in which I'll be focusing on this morning is going to be something that's called the innate system. This is a system that's with us all the time. We don't have to generate this part of the immune system like we generate antibodies, but it is a series of cells, phagocytes. I don't know if you've all seen My Big Fat Greek Wedding, but a lot of terms come from the Greek language, and phagocyte means a cell that eats. And so you have these cells in the body that eat up debris, that eat pathogens to get rid of them, to keep you healthy. And the one that's in the brain that does this is called a microglia. So I'm going to be talking about microglia a lot today. Then part of this immune response, this innate immune response, is this complement system. These are proteins, molecules, they're in your blood. Everybody has it. They're there all the time. And they are as a part of the first line of defense, saying, is this dangerous, and how is it dangerous? And they then, along with these other molecular danger sensors, proteins that say, this is something I don't expect, this is not good, they then send out, in collaboration with the cells of the immune system, send out signals, which we're going to call cytokines. And cytokines are like hormones, and they're produced by one type of cell, they're secreted, and they go throughout the body, and they hit other cells and say, okay, they're essentially directing how those cells respond so that you can have an appropriate immune response to keep you healthy. So these are some of the elements then of the immune system that we're going to be talking about. And we're going to focus on that innate system because that's where much, not all, but much of the inflammation comes from. And when we're talking about inflammation, and many people understand that as what happens in what I say is the periphery, outside the brain, in the periphery. And it's an immune response to a perceived injury or infection in which you get this heat, which is a fever, you get swelling, redness, because you actually have increased vascular permeability and part of your blood fluid and cells comes out from that system, and you get swelling and pain. In neuroinflammation, however, it's a little bit different because there's no, in general, there's no heat, no swelling, no redness, and no pain in general. But this neuroinflammation, which is essentially, I'll be telling you, derived from those cytokines and other toxic molecules, are in fact detrimental to neurological function. And in Alzheimer's disease, we're talking about detrimental to cognitive functioning. And so neuroinflammation then, one of the main components of that are those microglia that I mentioned earlier. And you get a proliferation and activation of those microglia, and that induces then these pro-inflammatory cytokines. Pro-inflammatory cytokines then are deleterious to neuronal function. So what are these microglia responses, and why don't we just get rid of all of them? Well, these microglia responses do indeed jack up inflammation, which is detrimental. That's bad. But the microglia also do good things. They eat cellular debris. They counteract infections that might occur. So those are good things. They're going to get rid of misfolded proteins that might otherwise damage neurons. And in addition, the microglia can indeed have reparative functions. They can remodel the synapses. So those are good things. So we're kind of in a dilemma here as we investigate this system. And that's what I would like to portray to you, is sort of the complexity of this. But we're making progress. And what we now know is that there are several different kinds of these microglia. Some are called classical activation, which is what people think about when things get activated. It's inflammation, which is bad. And that's produced by a particular cytokine, and it is an attack function. It's putting out really more toxic molecules for the brain system. On the other hand, there's this alternative activation, and that's stimulated by a different set of signals, these cytokines that have been produced in response, molecular responses to perceived danger by the immune cell systems. And that's anti-inflammatory. So, gee, that would be good, because that's not bad for the nervous system. And then there's this thing called acquired deactivation, and that's also stimulated by another set of cytokines. And these cells can be indeed phagocytic, a good thing, but they're also immunosuppressive. So that's good for a while, but you don't want to be totally immunosuppressed all the time. So the interesting part of this is that these cells, very different cells, can coexist all at the same time. And so what we want to find, and what we are finding now, because we now know that there's these different classifications, is that we can now look at these specific tools, these specific molecules, and say, oh, could we manipulate the entire environment by upregulating one set and downregulating another? And so this is the kind of thing that people are trying to do in terms of learning about inflammation. But again, we're at a relatively beginning stage, but we're making progress because we can now break down these different systems and identify molecules that then can be targets, or what they do, the next step down in the pathway can be a target for a very specific therapeutic that doesn't disrupt the good things, but only disrupts the bad things. So here's where I'm going to come to complement, and I mentioned this is part of the innate system. And complement is actually a series of 30 different proteins, again, that are in blood and in extracellular fluids, also on cells, and they are activated by infection or injury. And I'll show you this cartoon of sort of a schematic of the complement cascade. And obviously, there's not going to be a test at the end of this, and so don't get too confused. But I did want to say that there are lots of different components of this complement cascade, and that's not a bad thing. That's actually kind of a good thing. Once we can figure it all out, then we can, again, selectively go in and inhibit those things that we want to inhibit and allow the other things that are protective to be maintained. For example, these are the proteins that are very important in recognizing danger. And of course, the goal really of this immune system is to protect from infection. And so once they recognize it through these different pathways, you then trigger a series of effector roles, and one is that it enhances, these proteins can enhance phagocytosis, and that's a good thing because, say, there's this little bacteria in here, and it has now complement proteins deposited on it because of this activation event. A number of proteins can cut other proteins in half, which then allows them to bind onto the bacteria or other pathogen, and then the cells, the phagocytic cells can recognize it as, oh, that's dangerous, I should eat that, and they will then clear that particular pathogen. So that's one thing that the complement system helps on. The other thing is that with these other enzyme systems or cleavage of proteins, they send out new signals. One is this C5a molecule here, and it goes away from the area of injury, and it interacts with other immune cells and says, oh, we need help in here. Come in and help clean up this dangerous situation. And so C5a is a little sentinel that goes out and attracts in other cells that will be protective. And finally, this, which we won't talk very much about it, but this complement system can also assemble a pore or a hole in the bacteria, essentially killing the bacteria so that it will not proliferate. And then those cells that have come in to the area can then also not only kill any leftover live bacteria, but can also eat up those bacteria so that you don't have a bunch of bacterial carcasses floating around in your body. So that's a very nicely orchestrated system that has evolved. So these are all good things. However, it turns out that if this complement system is overactivated or not controlled, you end up by having many, many different diseases, many of which you all would be familiar with. Psoriasis is one, systemic lupus is another, but we also have age-related macular degeneration that is a major risk factor, is dysfunction of the complement system and actually the regulation of that complement system. And now what we would like to propose, and which I'll show you why we think that this is a good candidate for Alzheimer's disease. So let's go back now to Alzheimer's disease. And why would we think that inflammation plays a role in this? Well, we think one of the reasons for that is because you have, again, this is a human brain section. The brown here are the amyloid plaques. And these black wispy things are not just dust, but indeed are these microglia. And they're activated microglia, and they're all over these plaques. And so when in the periphery people see phagocytes surrounding a particular injury, that suggests inflammation. In addition, and I'll show you a little bit later, there's complement proteins on those plaques as well. And in the brain, scientists for a very long period of time have known that there are increases in pro-inflammatory cytokines in Alzheimer's disease. And most recently, as Frank just mentioned, these genome-wide associated studies have picked up several of these complement-related genes, complement receptor 1, clusterin, which is a regulator of the complement system, and then TREM2, which regulates phagocytosis, as being increasing the risk factors for Alzheimer's disease. And so there's just here again, this is this genome-wide associated studies. They show, they can find by sequencing the genes that there's single nucleotide changes in the DNA, or differences in the DNA of the individual, which alters the way these proteins work, perhaps in a slight way, but over time and with challenge, those become noticeable and increase the risk of disease. And these are just some of the risk factors that have been identified for Alzheimer's disease that relate to these processes of phagocytosis, induction of inflammation, clearance of misfolded proteins. Okay, so the question was, how would we recognize these different things? And first off, you can get your DNA sequenced so that you can find out if you have some of these polymorphisms. But we really don't, at this point in time, know how those differences are affecting the pathogenesis and the development of Alzheimer's disease. And that's what we need to know first, because then we can design and target, which is what we're trying to do here, is identify targets so that we can design therapeutics to abrogate detrimental consequences of those changes, genetic changes, or to kind of buff up where you're maybe lacking a little bit in a particular component or regulatory component. So we're finding out, just as we're finding out about PET scanning, we're beginning to be able to find out what things are available, but now how does that all work together, and then can we get a target? And so I'm going to tell you what we're doing in the lab at UCI that is sort of targeting this. And so I'm going to pass through this one and just mention why we think, or give you some hope that in fact starting at this point we might be able to see some consequences. So about 12 years ago, people in basic science labs were studying basic processes that are involved in other kinds of inflammatory disorders. So these are these other diseases that NIH is sponsoring research into. And what these molecular biologists found was that there are certain proteins in cells that come together after receiving certain signals of danger and form a complex that can cleave inactive cytokines to active cytokines. And that example is this pro-IL-1 beta, which is inactive. It's in the cell. It doesn't harm anybody. But once this inflammasome gets activated from signals, that gets cleaved, and now that is secreted out of the cell, and it causes the inflammation. And the things that could activate this inflammasome were things like cholesterol crystals in atherosclerosis, monosodium uric acid crystals in gout, and in type 2 diabetes. But now we also know that amyloid, fibrillar amyloid, can also activate this inflammasome. And so the good news about that is that when people 12 years ago found out about this inflammasome, they were able then, by knowing what the mechanism was and the regulation of this IL-1 beta, they were able to generate a target for whoops, I screwed up, wait. Okay, for this inflammasome, and were able then to actually treat people who had severe inflammatory diseases such as Mediterranean fever and other cold-induced inflammation, people that was very debilitating in 10 years. They were treating these people very, very successfully. Now, that's acute inflammation, so that's a little bit different than Alzheimer's disease, because Alzheimer's disease is over a long period of time, and treating and testing any drug takes longer because you have to look at it over longer periods of time. But you can go with this mechanism of identifying molecular paths, devising what the pathway is that is causing the disease, and designing a drug that is going to be able to ameliorate those symptoms. So it can be done. We just need to convert it here for Alzheimer's disease. So what has been done, just to update you on this, what has been done in some clinical studies of testing this inflammation. Do we have any evidence for this? Well, many years ago, maybe back in the 80s, people were doing some prospective epidemiological studies that suggested that NSAID use decreased risk or delay of onset of Alzheimer's disease. But then when we went and did other more direct, it was relatively low-hanging fruit. Treatment trials were done with anti-inflammatories, and they were without success because they were using really broad-based reagents that can cut down on all different kinds of things that are occurring in inflammation, the good ones and the bad ones. And in addition, similarly, prevention trials have had no effect. Again, because I would postulate, and others postulate, is that you have so many effects of this inflammation, you really need to have the specificity of these therapeutic targets is very critical. So we know that the easy stuff didn't work because it was a sledgehammer type of an approach. So perhaps we can look at some differential properties of peripheral inflammation, which these things we're addressing as well, versus neuroinflammation, and we may be able to see some differences there. So let me then tell you to go back now into the brain and try and walk you through some of the steps that, as a basic scientist looking towards getting a therapeutic out there, what kinds of steps we go through and why we think that this is a good thing. I should say a good candidate for looking at it, and part of those millions and billions of dollars that we really do need to be able to continue on. So as others have shown you already in Alzheimer's disease, you have these amyloid plaques. And as Dr. Flesher also said, many non-demented elderly also have amyloid plaques. Well, actually, even more people have amyloid accumulation in them than you can actually see in PET, because PET only sees one form of those amyloid proteins, and that's this fibrillar amyloid. And you can see this on a brain section once somebody is deceased by using particular stains, and that's called thioflavin, and that's this green thing here. And you can see that in the Alzheimer's disease patients that was diagnosed with Alzheimer's disease, they have these fibrillar amyloid plaques. So the amyloid is there, but now it's in a particular conformation. And you don't see that in these other plaques that are pretty diffuse. And that's going to be important because what we have seen is one of these complement proteins, one of the initiator ones that recognize that something is wrong or bad, it's called C1q, is actually able to bind onto these plaques that are fibrillar, but there's no C1q, and therefore no activation of this complement activation pathway in those people that don't have this specific form of the amyloid protein and are non-demented. So this correlated with doing things biochemically in the lab as well. So we knew we had a specific interaction. So our big model then, what the model that we're looking at, and when we're looking at this particular component of Alzheimer's disease that we would like to target to slow the progression of the disease, is here, that you get a damaged neuron. That's that initiating factor that we don't really know what all of those factors are. You know, traumatic brain injury could be one of them. But you then induce the production of this A-beta molecule. And as it turns out, then those A-beta molecules form these fibrils. In addition, when neurons are damaged, they begin to make more of these complement components, but early on in the disease, they really start making this C1q molecule, and only that C1q molecule, and I'll come back to that a little bit later if we have time. And they also, later on, when more and more fibrils are forming, so they're getting more toxicity in the brain, you get the induction of more of these complement proteins. Now you have the activator, these amyloid fibrils in the brain, and the brain has induced all of these complement proteins. Now you're going to get activation of the complement cascade, and that's going to generate many of these products like C5a here that's going to bind onto this cell, a microglia, which then says, oh, there's something dangerous here, so I need to put out a lot of these pro-inflammatory cytokines and other toxic molecules that can damage the neurons, and you get a cyclical effect here. So of damaging more neurons, and eventually these neurons are going to die. That's a really bad situation. So what happens then? Another thing that happens in Alzheimer's disease is not only do you get some of these pro-inflammatory cytokines being made, but as those microglia move toward and interact with the plaques, they actually interact with other danger signals, and there's another molecule here on the surface of this microglia that can recognize the fibrillar A-beta molecule and says, oh, this is really bad. And now the two systems synergize so that now you get a huge amount of these pro-inflammatory cytokines being produced. So the idea then, if that's the case, would be what if we knock out this interaction between C5a and its receptor on the microglia? You don't want to knock out all microglia because there's some good things, right? You just want to really pinpoint that interaction. Could then we decrease all those pro-inflammatory cytokines, decrease the amount of neuronal loss, and therefore slow the progression of cognitive loss in Alzheimer's disease patients? So we have to go back now and test this. And so what I want to show you is the pharmacologic and genetic evidence that we have that says that perhaps this is going to work. So first of all, there was indeed a small molecule that is an antagonist of this receptor C5a interaction that other people have shown in other, even neurodegenerative diseases, acts in a protective way. So it's a small molecule, and it interacts specifically, at least as far as we know, and many tests have been done, with one of the C5a receptors that is the one that causes the pro-inflammatory responses. Okay? So we took this, this molecule, we fed it to Alzheimer's disease model mice. And we have to use mice to begin with because we don't know if this is going to work. So we used mice, we treated them for 12 weeks. You can put this stuff in the water, they drink it up, they like it. I don't think they're getting high on it, but anyhow, they are doing this. So, and we gave this to them at the time of in this particular mouse model where they are accumulating amyloid. And then we looked in the brains of these. I'm going to go over this kind of quickly because it's, I think you can just see the general pattern here. In the untreated mice, you have a lot of green dots here, but in the treated mice, you have a lot less. The green dots are those fibrillar amyloid plaques. And if you quantify a lot of different mice and the load that they have, you get over 50% decrease when you treat these mice with this particular compound. In addition, this red in the next panel here is indicative of astrocytes, and astrocytes become reactive and when there's a lot of plaques around, and they can also be generating some pro-inflammatory cytokines that aren't useful. That amount of astrocytes also goes down in this treated animals, and the microglia, which we can stain brown in this panel, that amount of staining also goes down during the treatment. So you can get a 50% decrease in all of these indicators when these mice are on this particular drug. We had another mouse model because, let's try it in another group of animals that has a little bit of a different genetics. And so we used Frank's mouse model, this triple transgenic. And one of the good things about this mouse model is that you can see in the brains of these mice these hyperphosphorylated tau's, which is like the tangles and the plaques that are seen in human Alzheimer's disease. And when we looked at the tau in the untreated animals, all this brown stuff in the brain of the triple transgenic, you see a lot of brown staining, but treated with the particular drug, you get almost a 70% drop in that. So this was encouraging to us, and when we looked at the other markers in these mouse brains, like the fibrillar amyloid and microglia, they were also reduced as well. So that says, well, maybe this interaction between this molecule, so now we're into molecules, okay, and all of you guys are now molecular biologists here. We're into molecules. This molecule interacting with this receptor, and this is the little cartoon, this is the molecule over here, on the receptor, on the membrane of the microglia, that could be a target. Well, if you're you can't really go to a drug company, we tried, and say, oh, this is going to work, you should try this, and they're going to say, I'm not putting billions, or many, many millions, up to billions of dollars into this, if that's all the evidence that you have. So we went back, again, as preclinical studies have to do, and try and get more evidence for this. The idea is, well, what if we used a mouse, an Alzheimer's mouse, that didn't have that C5a receptor? What would happen then? You would predict, right, that the mouse would not get the disease so readily. And in addition, if we did another mouse model where you increase the amount of C5a being generated, you would accelerate that. So these are sort of hypotheses that we can test in these mouse models. And so we decided to also look at more behavior, because that's what we're interested in, right? If you have pathology and no behavior changes, who cares? You really want to see the cognition. So we focused then on this object location memory test for the mouse. And it just means that you put the mouse in here and you let them look around, and there's two identical objects in here, and they recognize that those are those two objects in there, and after a while they're not interested in it anymore. If you put a new object in, they're going to be more interested in it. Or if you change the location of one of those molecules, which you do in the next day, if the animal has memory, they're going to realize, oh, this has changed, this is new, and they're now going to be more interested and start looking at that object, and if they've remembered that. On the other hand, if they don't have memory, then they're not going to be able to discriminate that, and they're going to look at both of these objects at the same time. So if they're able to discriminate, if their discrimination index is high, then they're normal, nice mice, and if they cannot, that means they have something wrong with their cognition, which is what happens when in an Alzheimer's disease mouse model. So let me show you what we got on this test. Here is the Alzheimer's disease mouse model. Its discrimination index is very low. It's in even the negative. These are the wild type mice here, so they're discriminating just fine. And if you take the C5a receptor out of this mouse, you can genetically, not out of a particular mouse, but you breed the mice, okay, then that doesn't really affect their ability to distinguish this, to distinguish between the events. If you take this Arctic mouse model and you cross it to a C5a receptor knockout, as we call it, so they're now going to be devoid of that receptor, and you wait for 10 months, you now see that these guys here that don't have the receptor are much better at that cognitive task. So that says that without the C5a receptor, something is happening. We would say you're not triggering that inflammation. Something is happening in the brain that allows those animals to withstand. Okay, so the next one we did was we then overexpressed the C5a molecule by a genetic trick that we can talk about at another time. But you would then anticipate that you would accelerate the disease. In this Alzheimer's disease mouse model that I showed you before, they were 10 months old. Alzheimer's is an age-related cognitive loss, and in those mice, it's also their distinction index is lost with age. At 5 months of age, these red bars here, these Alzheimer's disease mouse models are okay. They can distinguish just as well as the wild type can. And even at 7 months of age, they're doing fine. But if you have now a mouse that has this Arctic mutation in it, so it's an Alzheimer's disease mouse model, but you also have it making much more of this C5a, you can see that their cognitive ability or their discrimination index is very decreased at a much earlier age. So both at 5 months and at 7 months. So this is then all consistent with the fact that these animals are that the C5a receptor is involved. Now let's go to one other different genetic mouse model. Again, the triple transgenic, because people are different and mice are different. Let's just see if it works again. And indeed, when we used Frank's mouse and we crossed it to this overproducing C5a model, we saw that the discrimination index at 16 months of age, these animals, they age differently, they develop their amyloid plaques at different times. At 16 months, these triple transgenics were doing just fine on this task, but when they were overproducing the C5a, they did not do well. Okay, this is exactly what you would predict. And indeed, when you looked at the pathology in these mice, they had much more fibrillar amyloid plaques involved as well. So they had increased plaques and decreased cognition relative to the other animals. So that says that C5a and C5a receptor is not the only thing that's going on, but it certainly is contributing. And perhaps then this synergy between this system and the fibrillar amyloid plaques can be altered such that you can inhibit then this robust microglia response. So we don't know if that's really the mechanism, and we're looking at that now to try and further define the mechanism, because it could be that this drug, this PMX-205, could be signaling on neurons, astrocytes, or endothelial cells, all of which are a positive thing. So we just need to make sure that we're not having side effects when we look at these different things. But it is saying that this drug is having an effect. We actually don't even know whether it's all occurring in the brain or whether there's peripheral inflammation that is also being suppressed so that you don't get those cytokines going into the brain. So we've got a little ways to go, but it's positive. So just to summarize this main part of the talk, amyloid deposits in AD brain activate the complement system. So you all know now what the complement system is. These inflammatory markers also increase in part as a result of this complement activation in mouse models of Alzheimer's disease. And perhaps targeted inhibitors of this complement system may be an approach to prevent and or slow the progression of cognitive loss in Alzheimer's disease. So I only have a few minutes, so I'm going to really skip over most of these slides, but I want to, again, try and put home to you the fact that we don't want to inhibit all of the complement system. First off, it's good for combating infections. But the other thing that we have also found, and I'm going to jump through this real quickly, so I just want to give you the bottom line here, is that remember I told you that C1q was generated early in the disease, and we know when in mouse models C1q is being synthesized, and it's early in life, actually before plaques come right here. And it's only later that the other components of the complement system come up. Turns out that this C1q actually has neuroprotective roles, and we've shown that this C1q by itself, in the absence of C5a downstream, is able to actually help clear cellular debris like dying neurons so that they don't cause inflammation. And it also reduces cytokines. So that's what you can see here. Everything below the line means that C1q is reducing the production of cytokines by microglia when only C1q is there. So that means, and we've done some other studies indicating sort of the mechanisms by which that does, by which the C1q is neuroprotective, and I'm going to pass on to this. But the point is that you don't want to knock out all the microglia, you don't want to knock out all of the complement system. You want to really go in with the scalpel and find out which ones are indeed the bad ones, and that's what's taking the time, and that's what's taking the effort on these different approaches. So when we know about these neuroprotective ones, then, well, gee, could we facilitate that? And so since C1q promotes neuronal survival directly, and I passed over that, perhaps we could use an inhibition of the C5a, but an enhancement of these downstream targets of C1q, this other complement components. Coming back to that main thing I said I was going to tell you about, which is a cocktail approach, in which you inhibit the detrimental events, but you promote the positive ones. And of course, these aren't the only elements of a cocktail, because there's many, many other targets, therapeutic targets for Alzheimer's disease, from secretase inhibitors, which people have not been able to find to work, but prevention of enhancing clearance, I should say, or blocking different forms of amyloid. But all of these together then should give a paradigm and give the tools, the therapeutic tools, that the physicians need to treat this devastating disease. So we're going from molecules to pathways to pathology to the patient, and we hope that we're going to get to the disease therapeutics, clinical trials, benefits to to patients. And I just want to also give you an idea of how many people. This is just my laboratory, my laboratory at UC Irvine. Lots of different people are involved in this. A talented graduate student, Tracy Cole, another postdoctoral fellow, Maria Benoit, who really put a lot of effort, as well as all of these other individuals, from undergraduates to graduate students to sophisticated, talented Marissa Fonseca is an immunopathologist, has done all the beautiful immunohistochemistry that we have. And people in the background, Sophie Chu, who manage all these mice, which actually is not as easy as you might think. In addition, then, our funding source, NIH and the Alzheimer's Association, major contributors, cannot do this. We just can't have people doing these experiments without the funding, so we're very grateful to that. So I will take questions. Okay.
I'm 87 years old and I feel perfectly fine. I have a little bit of short-term memory, but that's part of aging. Other than that, what should I do?
I'm sorry, what was the last question?
What should I do if I want to live longer? Just keep going? What?
Well, you want to avoid inflammation, keep your body healthy. I must say, you don't really want to eat a lot of Big Macs. I don't know if you do that,
No,
but
I
most
don't eat
people,
Big Mac.
Everybody's system is different. And that's one of those contributing factors that Dr. Flesher was talking about earlier, about how everybody's brain handles the amyloid a little bit differently. But clearly, those that have predispositions to inflammatory responses, which we saw in the genetic studies, are going to promote this. So I would say you're doing a pretty good job of adjusting your lifestyle to your genetics, because you're as good as you are.
Thank you.
Oh, more exercise is very good, but not to too much. Don't worry. Take a little bit of ibuprofen, when you get that tennis elbow and stuff. You keep the inflammation down. Okay. And I'm not a physician, so I really don't know. I really don't know. Okay, another person?
I've heard that brain exercise, such as playing certain types of card games, bridge and things like that, is helpful. Could you explain if it really is and why?
Actually, I'm going to leave that for Dr. Claudia Kawas, who's coming up here right after I get off, and she's going to explain it. She is the expert in all of that. And we'll give it to you in good terms. Okay.
Yeah, you distinguish between general inflammation makers in the body versus inflammation of the brain. And we've heard about books about decreasing inflammation in the body, et cetera. Are there any specific recommendations for reducing inflammation of the brain? This is fantastic, but as far as right now that people can do to apply that to that specific inflammation that we're targeting, diet.
People are still learning about what is it that is in fact about traumatic brain injury. You want to avoid that. So that's where people have the helmets. That's why there's all those concussion new rules and things like that. So you want to really avoid that. So if you want to go skiing, don't take that extra damage, but do you want to live that way? Everybody has to make their own choices on that. So I think that, damage, concussions or injuries to the brain are to be avoided. The other thing that's very important is that we do know that systemic inflammation due to bacterial infections also appears to increase the rate of Alzheimer's disease onset. So brush your teeth. Floss. And just in general, try and be healthy. One other question over here. Yes.
Yes, I was wondering, is a positive high C-reactive protein blood test a biomarker for this cascade?
Thank you. The answer is sort of yes and no. which is oftentimes the problem, okay? But the C-reactive protein definitely goes up with inflammation. Claudia Kawas is going to talk about those markers specifically, so I'm going to let her spend more time talking about that, if that's okay. And we can always come back this afternoon if you have any other questions, okay? Thank you very much.